To hear, smell, taste, touch, and see are common for everyone's daily activity, made by an animal, insect, or human. These senses are important by means of communication, eating, feeling, and seeing. But how do these senses work? So let's find out. First, in sensing things, we have what you call sense receptor cells, which can detect stimuli such as chemicals, light, tension in a muscle, sounds, electricity, cold, heat, and touch that will be sent to the central nervous system or the brain. When the action potentials is sent to the brain, the job of the sense receptor cells are done. Sensation is the action potentials received from the sense receptors that detected the stimuli, while perceptions is the interpreted sensation by the brain that can be a color, smell, sound,
Now,
or
we will
taste.
discuss how sensory receptors convert stimulus energy to action potentials. Our sensory organs, such as our eyes and taste buds, contain sensory receptors. These sensory receptors are specialized cells or neurons that detect stimuli, and these stimuli represent forms of energy. Meaning, when we say that the sensory receptors of the eyes detect stimuli, it actually detects light energy, same as the sensory receptor in our taste buds detects stimuli, which are chemicals such as salt or sugar. Sensory detection means that the receptor cell converts one type of signal or the stimulus to an electrical signal. The conversion here is called sensory transduction. The sensory transduction produces a change in membrane potential of the receptor cell, and this change is a result of the opening or closing of ion channels in the sensory receptor's plasma membrane. Before we proceed, we may remember, according to the previous chapter, that a membrane potential is a potential energy stored by the membrane that exists as an electrical difference across the neuron's plasma membrane. And so, I'll show you an example of sensory transduction. A sensory transduction at a taste bud. This is a taste bud, which can be found in our tongue. Take note, the taste buds on the left of the tongue have sensory receptors that respond to sugar, while on the right respond to salt. Thus, the brain can distinguish stimulus types. Anyway, a taste bud contains taste spore, the sensory receptor cells, synapses, and sensory neuron. The first step of its sensory transduction is the sugar molecules, which are represented by the green circles, first enter the taste bud through its taste spore. This second illustration represents a sensory receptor cell. The second step is the sugar molecules bind to specific protein molecules like this sweet receptor, which can be found on the membrane of the sensory receptor cell. This binding initiates a signal transduction pathway that causes the third step. Some ion channels in the membrane close and the others to open. Therefore, there's a change in the flow of ions. These changes in the flow of ions create a graded change in membrane potential, called a receptor potential. Unlike action potentials, which are all or none phenomena, receptor potentials may change. The stronger the stimulus, the greater the receptor potential. So stimulus is converted to a receptor potential, and receptor potentials usually result in signal entering the central nervous system. The fourth step is each receptor cell forms a synapse with sensory neuron. Receptor cell constantly secrete neurotransmitter, the blue circles, into the synapse at a set rate, triggering a steady stream of action potentials in the sensory neuron. Lastly, the fifth step, the sensory neuron sends action potentials. This left part of the graph shows the rate at which sensory neuron sends action potentials when the taste receptor is not detecting any sugar. And this one on the right shows when there are enough sugar molecules to trigger a strong receptor potential, which can cause release of more neurotransmitter. More neurotransmitter increases the rate of action potential generation that signals the brain that The sensory receptor detects a stimulus. The brain interprets the intensity of stimulus, like the level of sweetness, from the rate at which it receives action potentials. How come a cake tastes very sweet on the first bite, and as soon you eat more of it, it will already taste less sweet? Another, how come odor that is strong at first seems to fade in time? There is this sensory adaptation. Sensory adaptation is the tendency of some sensory receptors to become less sensitive when they are stimulated repeatedly. Receptors become less sensitive, trigger fewer action potentials, thus brain will lose awareness of the stimuli. Sensory adaptation keeps the body from reacting to normal background stimuli and prevents our nervous system from overloading of useless information.
We have specialized sensory receptors that detect 5 categories of stimuli.
First, we have pain receptors. Pain often indicates danger and usually makes animals to withdraw to safety. Pain receptors may respond to excess heat or pressure or to chemicals released from damaged or inflamed tissues. Histamines and acids are some of the chemicals that trigger pain. Next, we have thermoreceptors. Thermoreceptors in the skin detect either heat or cold. Other temperature sensors located deep in the body monitor the temperature of the blood. The hypothalamus keeps the mammal's body temperature within a narrow range. The hypothalamus is a portion of the brain that contains a number of small nuclei with a variety of functions. The hypothalamus controls body temperature, hunger, thirst, fatigue, and circadian cycles. Then we have mechanoreceptors. Mechanoreceptors are stimulated by various forms of mechanical energy, such as touch and pressure, stretching motion, and sound. All these forces produce their effects by bending or stretching the plasma membrane of the receptor cells. When the membrane changes shape, it becomes more permeable to sodium and potassium ions, and the mechanical energy of the stimulus is transduced into the receptor potential. Chemoreceptors. Chemoreceptors include sensory receptors in our nose and taste buds, which are attuned to chemicals and the external environment. Internal chemoreceptors include sensors in some of our arteries which can detect changes in the amount of oxygen in the blood. Osmoreceptors in the brain detect changes in the total solute concentration of the blood and simulate periods when blood osmolarity increases. Electromagnetic receptors. Energy of various wavelengths occurring as electricity, magnetism, or light may be detected by electromagnetic receptors. Many animals appear to use Earth's magnetic field to orient themselves as they migrate. Last, we have photoreceptors. The eyes are probably the most common type of electromagnetic receptor. Photoreceptors detect the electromagnetic energy we call light, which may be visible or ultraviolet parts of the electromagnetic spectrum. A variety of light detectors have evolved in the animal kingdom. Despite their differences, however, all photoreceptors contain similar pigment molecules that absorb light.
Ears are used for hearing, and an important factor especially on communication. It has 3 major parts: the outer, middle, and the inner. Let us start first on the outer ear. The outer ear consists of pinna, auditory canal, and the eardrum. Pinna is the fleshy structure we humans call ears. In the pinna, there is this hole which is the channel or passageway called the auditory canal, and at the end of it is the eardrum, which separates the outer ear from the middle ear. The middle ear consists of the following: the hammer, anvil, stirrup, and the oval window. The eardrum, which separates the outer ear and the middle ear, is connected to the hammer, which is connected to the anvil, which is connected to the stirrup, and which is attached to the oval window. The inner ear consists of the cochlea, a fluid-filled coiled tube